Keller, and I'll be talking about one of the projects that I finished in my postdoc with Sujini. I'm sort of now in that ephemeral, figuring out what I want to do when I grow up. But so climate is something that's really important in ecology, and you hear about it all the time because it's increasingly rapidly changing in various areas. So, you know, hotter areas are getting warmer, or really cold areas are also getting warmer. This is particularly important in plants that have very limited dispersal. They basically have one shot to go wherever they are on the wind or whatever, and then they're kind of stuck where they are. And recently, epigenetics in plants people have gotten really excited about that because that's another phenotype that people hadn't really thought about for a long time that can modulate expression. This might be another way that you can get sort of a closer fit to the environment. And in plants, it's really important because, so here I'll actually define epigenetics. People talk about it in a lot of different ways. But what I'm going to be talking about is defined as molecular mechanisms that can alter gene expression and ultimately the phenotype without actually altering the DNA sequence itself. And so there are several different kinds. I'm going to be talking about DNA methylation in this system. And what's cool is that, so in animals it's really different where there's essentially methylation is reset early on in development. But in plants, it's actually heritable. People have shown this a bunch of different times. This is kind of the classic example everybody uses. So in Linaria, people had identified that there's these two wild phenotypes you can go out in the field and see. So the wild type displays bilateral symmetry, and then, but there's also this mutant phenotype that actually has radial symmetry. And what people went out and did is they went and looked at it, and if you look at the actual genetic sequence, there's no difference between these two morphs. And so what's actually driving this difference is DNA methylation that's binding in the open reading frame as well as the promoter region being 500 to 1,000 bases upstream of the reading frame. And so in this mutant phenotype, there's lots of methylation, and that basically represses methylation expression in this LCYC gene. It almost never works as cleanly as that, but it's a really good example. So this is kind of a precursor study by one of my collaborators, Jesse Lasky, and there are a bunch of other studies that have shown this, but we now know that there's a substantial portion of genetic variation in Arabidopsis that you can attribute pretty cleanly to climate. So in this first, so this is across the native range of Arabidopsis, and what they did is that there's some statistical tools that you can use to partition the amount of genetic variation that is due to space, and then because Arabidopsis has a very good association with isolation by districts, and so you can determine how much genetic variation is due to just population structure and how much might be due to climate. So climate is this kind of ephemeral conglomerate of 50 or 100 different climate variables. And so what they showed is that if you look at sort of the synonymous and non-synonymous mutations, they're actually quite enriched compared to the null expectation of sort of 1,000 or 100 shuffled genomes, essentially. And so this is strongly suggestive that there's a lot of this genetic variation that associates with climate and likely has some functional benefit. Now we're actually getting to the methylation part. So I was using two different studies in this. So one from Schmitz et al., who's now a new faculty member up at UGA, and then the second study is from Dubin et al., that's from Magnus Newell's lab. And so it's the whole genome is fully bisulfite sequence. So So basically you get, for each individual base, you get estimates of how methylated or not methylated is it. And so the other tricky thing about in plants compared to animals is that there's actually 3 different methylation contacts that are really important, and I'll talk about this later. But they're positioned differently across the genome, and they also tend to localize. So CG methylation, you tend to find it in gene body regions, whereas CHH tends to be in intergenic regions, and is often clustered around the centromere. So just know that there's different contacts, and they end up different places. And then so the other final thing before I actually start talking about what I did is that there's this concept of differentially methylated regions that people talk about in epigenetics, where so methylation is not just an isolated phenomenon. Like if you look around surrounding regions of some focal base, you tend to see that if one base is methylated, often the other surrounding bases are also methylated. And this usually crops up, so when you see genes that are turned on or off, it's often like the entire promoter is highly methylated or highly unmethylated. And then, so the climate variables we got from this really nice data set called World Climate. And so there's a really nice, easy to use interface in R that basically, so if you have latitude and longitude for any site, you can use this interface, and it goes down to about 1 kilometer base region. So there's really fine resolution to give you estimates of, this is really what we think it's like for plants to be growing in these regions. And all the red dots are just the collection sites for climate that was then sort of interpolated across the last 50 years. So it's a really fine scale map. It's pretty awesome. And then, so the actual statistical machinery we were using in this is called redundancy analysis. So basically the data that we're using is that on the sort of Y variables, it's actually sort of multivariate. So we have 42 different climate variables on the Y as sort of the Y variables, and then the X variables are either biallelic SNPs or differentially methylated regions. And so redundancy analysis is a way to, so you can think of it as a multivariate analog of principal components analysis. And so one of the things that's nice about it is that it deals with climate variables that tend to be collinear. So, and that's sort of a known problem in climate variables, is that a lot of them, so if you look at temperature in May versus temperature in June, it's going to be really similar. So this is a way to in some ways, kind of deal with that collinearity. And then also you can do a proxy of population structure using principal components of neighborhood matrices. And so this was all used with the vegan package in R, which is also pretty great. So this is just a visualization of what I was talking about. So you have all these different accessions, so 122 or 148 depending on the data set, and then you have your sort of Y variables like annual temperature, precipitation, where you break it down by month, or you do some other sort of combinations of them. And so we ended up using 42 different ones in this, and then you have sort of the genetics. So we did genetic and methylation genomes. It was about 150,000 sites, depending on the data set. And so then some magic happens, but then, so one of the things that you can get out of this is these climate gradients. That's the color in blue for the sort of single most important, or the one that has the highest loading. So like PCA, you get sort of the axis 1 of variation, axis 2. And so what I've shown is just isothermality and annual precipitation. And so these were the individual climate variables that had the highest loading, first axis of variation. so basically you just see that, and then the coloration of the dots is simply this population structure variable. And so you see that there's this east-west gradient in Europe, in the Europe data set, and then in Sweden, that second data set, there's sort of a north-south structure. But then there's also the actual climate variable doesn't cleanly sort of associate with that structure. So it seems like there's sort of different things that are contributing to this variation. And so another way to look at this is, so if we sort of dive down into the individual methylation context. So CG tends to be what you find in gene bodies, and then CHG and CHH tend to be found in other areas. And so there's this overenrichment in the first RDA axis of CHH. It's much higher than any of the sort of random genomes that we look at. As I was kind of hinting at before, these CHH methylation sites that are highly associated with the first axis of variation, they're found in intergenic regions or transposable element regions. And so that was in Europe, and then we also see the same thing in Sweden. And so now this is the last little bit of data that I'll show is, so if you actually look at the first three axes of RDA variation, it actually segregates with the methylation context again. So what I've shown is that on the x-axis is just the cumulative quantile of RDA1. So basically on the left axis are all the highly associated methylation sites, and then on the y is just how much of each context is found. And so basically what you can see is CHH is loading strongly on the first axis, and then CG is loading on the second axis, and then CHH is loading on the third axis. You see the same basic pattern in the Swedish panel. And then, yeah, I talked too much. But so in summary, I'll just say that, there's these two really different data sets that show the same basic patterns that we were talking about, where they're associating, so methylation is associating with climate, and you can divide it down into these different methylation contexts. And then DMRs tend to be important. And then, thanks. And then it's what people have been talking about.